So imagine you're trying to tune um, a high-performance car engine, right? But you're only allowed to study a single bolt on a completely sterile workbench.
Yeah, you'd have like zero concept of the actual vibration when the engine is firing,
Yeah.
or how the thermal dynamics affect the parts next to it.
Exactly. And for decades, that's kind of exactly how preclinical oncology trials have operated.
Yeah.
But today, for our deep dive, we are looking at KU Leuven's Trace platform, and they're basically abandoning that sterile workbench to watch the whole engine run.
Right, because historically the failure rate for oncology trials has been just massively discouraging. Traditional lab models simply fail to capture how complex a tumor really is.
And Trace is bridging that gap between, the limitations of lab tests and actual patient reality. So the cornerstone of their whole approach is this biobank they have.
Yeah, it's pretty incredible. They have over 180 patient-derived xenograft, or PDX,
Yeah.
models across 10 different tumor types.
Okay, wait. For those who might not know the term, a PDX model is basically where they take fragments of real patient tumors and transplant them directly into living, especially bred mice.
Exactly. And the reason this is a game changer is because traditional immortalized cell lines, well, they rapidly lose their spatial architecture, they lose their diversity.
Right? They just become a homogeneous clump of cells.
Yeah. But PDX models keep the whole picture intact. They preserve the original tumor's genetics, the cellular diversity, and really importantly, the microenvironment.
Which makes them vastly superior predictors of how a real patient will respond to personalized treatments.
It's the difference between just reading a recipe and actually baking the cake. You have to see how all those complex ingredients interact in a living environment.
That's a great way to put it. You need the oven to see how it bakes.
But I do have to push back a bit here because the science relies so heavily on live mice. And there is a lot of pressure right now to phase out animal testing entirely. So how does the center ensure this research is actually ethical and sustainable? Well, it might seem counterintuitive, but that regulatory pressure is actually what's driving the precision. Trace operates under really strict European and Belgian legislation. It's all anchored by the 3 Rs.
Right, which are reduction, refinement, and replacement.
Exactly. So for reduction, it means they only use the absolute fewest animals possible to get statistically significant data.
And when they do use them, that's where refinement comes in.
Right. Refinement is about optimizing the experimental design to limit any suffering. So they use state-of-the-art anesthesia, they use social housing in these ventilated cages, and they provide environmental enrichment.
So animal welfare is just central to the strategy. It's not an afterthought.
Not at all. It's a mandate for ethical, sustainable oncological research. But, even with perfectly refined PDX models, there is a glaring biological catch.
The immune system.
Yep. Once you engraft a human tumor into a mouse, it loses the human immune microenvironment.
Because it's relying on the mouse's immune system, which means if you're trying to test modern immunotherapies, your data is basically useless.
It is a massive roadblock. But to solve that immune gap, Trace integrates human models. They use new approach methodologies, or NAMs, to bypass the limits of animal testing.
Oh, wow. So how do they actually do that?
Well, they develop humanized in vivo PDX models by actually engrafting the mice with human hematopoietic stem cells.
Wait, really? So they are actively reconstituting a functional human immune system inside the mouse.
They are, which lets researchers observe human T-cell responses against the tumor in real time. And on the ex vivo side, outside the body, they use patient-derived tumor fragments, or PDTFs.
So instead of breaking the tissue down, they culture intact fragments of the human tumor, which retains the patient's native immune cells and the exact spatial architecture.
Exactly. So EMS and animal models aren't competing approaches here. They work in synergy.
Right, so the PDTFs handle the rapid specific immune testing, and the living PDX models validate the complex systemic behavior.
Yep. It gives scientists this ultimate toolkit to develop truly personalized cancer therapies based on reliable, translatable science.
It's an incredible paradigm shift. It really bridges the gap between theoretical efficacy and actual patient survival. But it leaves you with a pretty compelling question to think about.
Oh, definitely.
As these new approach methodologies continue to advance, will the replacement aspect of the 3 Rs eventually eliminate the need for animal testing entirely? Or, going back to our engine analogy, will truly understanding the whole picture of cancer
Yeah.
always require a living organism?